and good morning, Professor Tishman. Again, we have a webinar series through the Amity Institute of Nanotechnology, Amity University, Noida, and we are lucky that we have the Dr. Tristan Clement from the USA, who is our collaborator, as well as he is the guide of one of our PhD students. And he is associated with the Amity since I think 2015 or 2014. And after that, he is associated with that. So Dr. Tristan Clement completed his PhD study in 2014 as a part of the Bio-Nano Research Group led by the Iyer Swaminathan at the University of Western Australia. Following this, Tristan was successful in obtaining a NHMRC MRC Peter Daughtry Australian Biomedical Research Fellowship with the Finolo Wood Foundation research team, where his work involved investigating nanomaterial application for wound healing and scare treatment following burn injury. Tristan is currently an American Australian Association postdoctoral research fellow in Chicago at the Northwestern University in the laboratory of Professor Samuel Stuff, who is a very well-known professor all over the world. And Tristan has won a number of the awards, including the Exxon Mobile WA Student Scientist of the Year of his PhD work, a WA Young Tal Poppy Science Award, Australian Institute of Policy and Science, and recently was acknowledged by the WA Business News with the 40 under 40 People Choice Award, and as a CSA Science Finder Future Leader of Chemistry in 2018. Apart from that, he is quite active in the games. He is the international player of the hockey. He represents the Australian team in the World Cup as well as the Olympic. And he was also a part of the Indian Hockey League team. He play for, if I am right, Dr. Tristan, he play for Punjab.
That's right, Punjab Warriors.
Punjab Warriors. And we have the interaction. He played few matches in Bhubaneswar, in some Chandigarh, and from Delhi. So with this, I would invite Professor Clemens to start the lectures. Clemens, you have the around 45 to 50 minutes, and after that the question answer session. So now session, you can start.
Perfect. Thank you so much, Professor Sinha. I want to talk today about multifunctional polymeric nanomaterials for therapeutic applications. And my talk today is a collection of 3 different stories that I'd like to share around cancer treatments mainly. So nanoparticles for cancer treatments, as well as some interesting study that we've just finished here in the US looking at polymeric production. So using controlled radical polymerization, which I'll talk a little bit about. So before I get to that, last time I was in Amity was in 2017, as you can see from this image. And as Professor Sinha said, we also were here mainly for hockey. So here I am with Professor Sinha and Sardar Singh, obviously a very famous hockey player in India, before I presented this day. One thing that I was very happy with my very first visit to Amity was these images, and I was looking back through these. We played a hockey match between the staff members and the students in the sports department, and I'm glad to say that the staff members won. And hopefully, Dr. Sinha is the trophy. There was a trophy for this. Hopefully, that's still available somewhere at the campus.
Yeah.
So today, I want to talk a little bit about nanotechnology and how I've used that for a range of different treatments, mainly based in cancer. And when I was a young PhD student, I was very impressed by Richard Feynman. And Richard Feynman made this keynote presentation, I guess, at the the American Physics Society in the 1960s, 1959, where he said that, why don't we try and write the entire 24 volumes of the encyclopedia on the head of a pin? And I think what was so exciting for me about nanotechnology as a science is that the instrumentation, the methods that we can use to be able to see and actually image things on the nanoscale have been very, very new. So transmission electron microscopy was invented in 1933. So this was the first time we could actually see with greater resolution than light. To be able to actually image atoms, so to be able to see individual atoms, wasn't until the scanning tunneling electron microscope was developed in 1970. So this is only 50 years ago that we're able to then see individual atoms. And then the AFM, the atomic force microscope, which is a very useful instrument for seeing on the nanoscale, wasn't invented until 1980. So this is only 40 years ago. And so for me, thinking about this as a science, thinking about nanotechnology as a research space, we've been doing medicine for hundreds and thousands of years. We've been engineering bridges and castles and forts for hundreds of years. But we haven't been able to see and know what's happening on the nanoscale for at least the last, 100 to 50 years to be able to see these things. And for me as a researcher, that's very exciting because there's lots of room for discovery in this space. So nanotechnology, this is just a word cloud, but I just wanted to highlight that even though it's a very new science and a very exciting space to be working in, a range of products have already made their way into the market. If we think about in the top left here, you can see here these are different sunscreens that are now infused with nanoparticles to absorb UV light to protect our skin. There's transition lenses, so in spectacles and glasses that they have silver nanoparticles in the lenses so that they're photochromic, which means as the UV light hits the lens, they're able to become darker to actually get a better tint to protect your eyes. We can make materials such as Gore-Tex, which is a fabric they use for clothing, or surfaces, hard surfaces like this one here, that are super hydrophobic. And in saying that, they're able to repel water very well and stay clean or actually stay dry, for example, in the case of Gore-Tex. And if we think about high-end sporting equipment, so Rafael Nadal, the tennis player, he's been working with a tennis racket that's been infused with carbon nanotubes to have a much stiffer tennis racket, as well as having a very light tennis racket. And bike frames now, so high-performance bike frames that they use in races, such as the Tour de France, for example, are now starting to be infused with carbon nanotubes to have a bike frame that is much more stiff, but is very, very light to actually allow them to ride faster in the races. And finally, this one on the left here, nanotechnology is making a really big push into nanomedicine and nanobiomaterials. And today, that's what I wanted to speak about in my presentation. So I want to talk about 3 stories today about polymers and drug delivery. The first one is looking at an in vitro study, so a cell-based study looking at the active and passive targeting of nanoparticles. And this was work that I did at the University of Western Australia just before I left to target cancer cells, and seeing if targeting the cancer cells or just having nanoparticles taken up by cancer cells had a different effect on their toxicity. I want to talk about a very recent study we've just finished here in the US at Chicago, where we're using quantum dots for photo RAFT polymerization, and I'll talk a little bit about that and why RAFT polymerization is very useful as a technique. And then finally, I want to talk about using this RAFT polymerization to form nanoparticles. So PISA, P-I-S-A, is an acronym for polymerization induced self-assembly. So these nanoparticles assemble on their own and being able to use those as a way to kill cancer cells, for example. So these 3 stories will make my talk up today. So the first one, as I said, came from the University of Western Australia. There's many people that have worked on this project with me, and this is my supervisor when I was at the University of Western Australia, Swaminatha Iyer. Essentially, the concept of this study was very simple. We wanted to take a polymeric nanoparticle, and I'll show you these on the next slide, and we wanted to have one where it had a peptide on the surface that actively targeted receptors on the cell surface of a cancer cell. And we then wanted to compare the passive uptake, so taking a nanoparticle that did not have the targeting ligands, the targeting peptides, to see which one had better efficacy in a cancer model. So Swaminatha Iyer here was my supervisor at the University of Western Australia. And we made these nanoparticles throughout my PhD and my first postdoc in Western Australia, where essentially we could take a very tiny nanoparticle. You can see here from TEM that they are approximately 100 nanometers in diameter. And we can then load things with inside this polymer nanosphere. We can add fluorescent dyes, so they're fluorescent, so we can see them and use them for cellular imaging. We can add magnetic nanoparticles inside the core of these, and we can then use them for MRI, so magnetic resonance imaging. And we can also then add therapeutics into the core of these molecules, into these particles, sorry, and have release of that drug at an injury site. So for the work I'm describing today, we've actually loaded inside these nanoparticles a chemotherapeutic being docetaxel. So looking at these nanoparticles very quickly, we can functionalize the surface of them to have a highly positive surface charge using a polymer. And this high positive surface charge, this PEI, so polyethyleneamine, allows for these nanoparticles to get inside cells. So if we look at these microscopy images, when we have the PEI, so when we have the PEI on the surface of the particles, they associate very quickly and rapidly with cells. When we do not have the PEI, we can see we don't have that cellular uptake with these nanoparticles. The video on the left here is just a time lapse which shows that uptake over time. And then if we looked at the TEM, so transmission electron microscopy again, we can see that our nanoparticles are actually engulfed by the cells. So this is a macropenosome, which is coming up and folding over and actually bringing that nanoparticle here inside the cell. And you can see in this image, we have a number of nanoparticles getting taken into the cell and then packaged together. So we wanted to look at comparing this idea of passive versus active uptake of nanoparticles by cells. And this is something that's a lot of debate, you could say, in the nanomaterials, nano drug delivery world. And Warren Chan, a very famous professor from, he's in Canada, was saying, and he looked across all the different nanoparticle formulations that they've used for cancer treatments and showed that there wasn't a huge difference between passive uptake and then targeted active uptake. But they did see a little bit of improvement in terms of the targeting strategy using an active pathway. And so when I say passive and active targeting, I wanted to just highlight that for you here. So this is a paper that came from Robert Langer's group. So Bob Langer is obviously a very famous researcher, and Dan Peer, a very good researcher out of Israel. And so the difference between passive and active uptake is if we have this here, this is simulating blood flow and a cancer tumor that is growing on the side of that vasculature. We can see that if passive nanoparticle uptake is through essentially this leaky vasculature here, allows the nanoparticles just to seep in. They can go through the gaps in those junctions and find their way into the cancer tumor. This is known as the enhanced permeation and retention effect. Where active cellular targeting is when we actually put a surface on the nanoparticle that then wants to actively bind to the cancer cell. So if we look inside this box here, for example, if the nanoparticle has in blue these ligands, which in my case will be a peptide, it can then interact with these cell surface receptors, and those cell surface receptors can then take that nanoparticle into the cancer cell. So the concept of this study was to simply look at ways, how can we compare that active pathway versus that passive pathway? And so the ligand that we chose to target in cancer cells is this epidermal growth factor receptor, EGFR, in cancer. And this is highly expressed and overexpressed in a number of different cancers. So non-small cell lung carcinomas, in 80% of the cases they find in humans, this epidermal growth factor receptor is highly upregulated. Colon cancer, cancers of the head and neck, they show a high expression of this EGFR receptor on the surface. So we wanted to use that to our advantage. And we did that by making this peptide here. So this is the peptide sequence. As you can see, this is called the GE11 peptide. And what this peptide does is it interacts very strongly with the epidermal growth factor receptor. So if we can decorate, if we look at this schematic, this is our nanoparticle now. We have docetaxel, which is the chemotherapeutic, so the cancer killing agent within the core of the nanoparticle. And then we can then tether to the surface of this GE11 targeting peptide. And to do that, we just use this small linker with a cysteine residue and then did melamine cysteine chemistry on the surface of the particle to attach it. So now we have this nanoparticle which can have a therapeutic targeting sequence on the surface, and we can easily not put that on the surface to have the control particle of passive uptake. So looking at some characterization, these are what the nanoparticles look like by TEM. We can see again, they're approximately 100 nanometers in diameter. This is the dynamic light scattering showing that the targeting ligands, so when we have them with GE11, without GE11. They're about approximately 100 to 150 nanometers in size. There's a slight difference in the surface charge of the particle when we attach the peptide, but that's to be expected because we're attaching this very hydrophilic surface charged peptide to the surface. In both cases, the docetaxel, so the chemotherapeutic, can release over time from the interior of the particles. And in this video, we can see that this is a confocal, high-resolution confocal stack. The nanoparticles are labeled in red, and the cytoplasm is labeled in green, and the nucleus of the cell is labeled in blue. So we can see that the nanoparticles are localizing within the cell membrane. So looking at more of those images, you can see very clearly that in both cases, the GE11 targeted nanoparticles, so with the active uptake, are able to find their way inside the cells very well. And then the non-targeted nanoparticles, so without this peptide ligand, are also finding their way inside the cell as well. So this was interesting for us. We really expected the targeting particle to get into the cell better or at a higher extent than the non-targeted particle. But what we actually saw was that they both seemed to be taken up by the cells almost at similar levels. So we wanted to characterize this a little bit more. And so now we're looking at two different cancer cell lines. So this is the MCF7, it's a breast cancer cell line. And this MDA-MB231 is a different type of breast cancer cell line as well. And we're now measuring the IC50 value. So what concentration of our therapeutic nanoparticles is enough to kill the cells to 50% of their viability. And you can see here in cell culture that the docetaxel is very, very low. So, and that's to be expected. This is a very toxic therapeutic. It can actually kill the cells very, very well. So you need a very small concentration of this to get 50% viability. Compared to our targeted nanoparticles, so the GE11, we can see that it's also quite low. And there's no significant difference here in the MDA-MB3 between the targeted nanoparticle and the free docetaxel. But what we did see was that our non-targeted samples had much higher IC50 values, which tells us that the non-targeted particle was much less lethal than the targeted particle, which was interesting to us because you could see from the confocal imaging before that the nanoparticles seem to be taken up by the cells to the same extent. But if it's less lethal, that tells us that maybe the nanoparticles, depending on their pathway of being entered into the cells, is actually dictating how toxic they can be and the effect they have. And so we wanted to, in fact, test this a little bit further. And so now we're looking at just the non-targeted in the white, and in the gray is the targeted GE11 nanoparticle. And you can see here when we label the cells for endosomes and lysosomes, so two different ways that cells can clear debris and clear foreign bodies from their cytoplasm, we can see that the non-targeted nanoparticle in both cases has a higher persistence of being co-localized with the endosomes and lysosomes. And so this tells us that the nanoparticles that are non-targeted are being packaged up by the cells and encapsulated by the cells to clear them out, to actually get rid of them from the cellular cytoplasm. Whereas the particles that are going in by an active targeting mechanism, so these gray bars, are much less. And that tells us those nanoparticles are actually being accepted by the cells to a certain degree. And checking this in one other way, we thought of a very experiment to probe this a little bit further. So here we then used an antibody which could then protect on the cell surface. So, sorry, I should mention that you want to watch my camera because I also will do hand things as well as I speak because I'm not there in the room with you all. But in this case, we took an antibody which is able to block those cell receptors. So if this is the cell, for example, and all those receptors are on the surface, we took an antibody to block those. And by doing that, we would expect that the active targeted nanoparticle is then unable to find those receptors and then unable to actually get into the cell through that active pathway or that active mechanism. And we can see that when we have the untreated sample and the antibody incubated with the cells, there's no real change in viability. If we then incubate these with the targeted nanoparticle and then the non-targeted nanoparticle at their IC50 values, we see approximately 50% decrease in viability, which is what we'd expect. What was interesting though is that when we have the antibody in there So if we pre-incubate it and block these receptors, we see there's a rescue in terms of the actual efficacy of these nanoparticles in killing cells. And that actually is very good evidence for us that the active pathway, the active uptake, is through this EGFR receptor. Interestingly though, the antibody had no effect on the toxicity of the non-targeted nanoparticle. So this bar and this bar are the same height, for example. And that tells us that the uptake for the non-targeted nanoparticle actually is through a passive pathway. And so taking all this data together, we can then summarize that we have these targeted nanoparticles for cancer therapy, which, depending on their uptake and the way they're actually targeting the nanoparticle or targeting the cancer cell, can actually change how it actually interacts with the cell. And we then looked at ways that we could modulate this internal cell targets of nanoparticles, because this could be really useful for us to know about how we can actually engineer these nanoparticles to have the most benefit in terms of being able to kill cancer cells. And this EGFR receptor is actually overexpressed in a range of different injury models. And so the ability to not only target cancer therapy is very promising for this as a platform technology to then look at using this EGFR targeting to target other cell types, for example. And so this was recently published. If you want to see more of the details in Langmuir a few years ago, 2 years ago now. Okay, so with that, we'll move to the next story in the presentation. And this is to look at making polymers now with quantum dots. And this is probably a little bit more of an educational story. So this would be a good one for the students as well. This was some work we've just published here from Northwestern University in Chicago with a very talented PhD student, Kevin McClelland. And what we were trying to do is use quantum dots now to do RAFT polymerization or controlled polymerization. So really quickly, if we think about what is radical polymerization or controlled radical polymerization, we have free radical polymerization, which makes a lot of plastics and polymer materials that we have all around us, from phone covers through to safety glasses to laptops to desks. All of those materials are made with free radical polymerization. What controlled radical polymerization gives us is a range of different features. And this is now talking about techniques such as RAFT, which I'll talk about today, but also ATRP, so atom transfer radical polymerization, or nitroxide mediated polymerization as well. And what these methods give us is a way of being able to actually control what polymer molecular weight we get. We normally can get very narrow molecular weight distributions, where with free radical polymerization, we have no control over that polymerization process. And so the distribution of molecular weights is very, very broad. Where with RAFT and ATRP and these other techniques that are controlled, we can actually make those very narrow distributions of polymer chains. And so the way that RAFT works, so this was discovered in Australia, which is why I'm so excited about RAFT as an Australian. But we take a small initiator, a small amount of initiator. The M here represents monomers, and we can then initiate a vinyl monomer to start this polymer chain growth with a radical. So the small dot here represents a radical. The real key for RAFT, reversible addition, fragmentation, chain transfer polymerization, is that we have these dithio agents, or RAFT agents, as most people refer to them, okay? And these RAFT agents are very special in that they have these dithio groups, that when this radical polymer chain growth actually interacts with the RAFT agent, it can then stabilize and hold this radical very easily. And what this RAFT agent allows for is this equilibrium here to get established, where we have the polymer chain and the RAFT agent sitting together. And when it's on the RAFT agent, it's in this dormant state. So right now, in this middle component here, there's no polymer chain growth occurring. But when it shifts to this side of the equilibrium, then the RAFT agent passes the radical onto the monomer. Monomers can add to the polymer chain. It can then go back to this dormant state. And in doing that, with the RAFT agent, this dithio group being able to control the shuttling of that radical back and forth, this equilibrium, it's then able to ensure that most of the polymer chains grow at the same rate. And that's very important because that allows us to have most of the polymer chains at the same molecular weight. So one thing that I've been working on very fiercely here in the US is looking at ways that we can now use light. Instead of here where we might add an initiator, So those who've done polymer chemistry before might have used things like APS or other thermal initiators that degrade and make radicals with temperature. Now we're using light to be the initiating source. So here we can use very cheap LED lights and use visible light, so light that's around us, for example, to start this process off. And if we look at this equilibrium now, PC represents the photocatalyst. So we have some molecule in here, normally a fluorescent dye, for example, some molecule that can, when visible light hits it, it can be excited. And when it's excited, it can then interact with the RAFT agent and fragment that to then start this polymer chain growth and start this equilibrium here. Okay. And then it can go back to its ground state. And then with more light, we can then excite the photocatalyst again and repeat this process over and over again. And so this is a really nice method because it can allow for polymerization to occur at room temperature in aqueous systems, so with water as the solvent, which is quite difficult with other polymerization methods. We can use visible light. It's a very biocompatible, bio-friendly and green process. And it was also discovered in Australia, so I'm very fond of photopolymerization using RAFT technology. So when I first came to the US, we decided to think about, well, can we use quantum dots? So cadmium, selenium quantum dots to actually act as the photocatalyst in these systems. And so again, very similar to the schematic I showed on the last page, but here we have a quantum dot where light hits the quantum dot. It can then go to this excited state where we have a separation of the electron and hole pair. And in doing that, we then have an electron that can then transfer to the RAFT agent. And again, we have this standard RAFT equilibrium. Okay. And so I just wanted to show a little bit of data to represent this, and this is from the paper, but it also illustrates some of the advantages of controlled radical polymerization. So the first thing here that's very interesting about controlled radical polymerization is if we think about, first of all, that here we have time on the x-axis and we have conversion on the y-axis. So this is taken with NMR, and all we're looking at is the concentration of the monomer initially relative to the concentration of the monomer over time. And as a polymerization proceeds, the amount of monomer that we have in the system starts to decrease because it's converted to polymer. And we just wanted to show here that when we use the quantum dots, so the blue ones here, the quantum dots, this is a much steeper straight line. The fact that it's straight and linear is very important to show that it's a controlled polymerization process. The fact that it's much steeper relative to eosin Y, which is a fluorescent dye they commonly use for this, tells us that the rate of this reaction is much more rapid when we use the quantum dots relative to using an organic photocatalyst. One thing about using light as your initiation source is it's very clever. You can actually then turn on and off the light. And in doing that, you can then turn on and off the polymerization in your process. So in this graph, we have the off is in the blue regions and on is in the white regions, you could say. You can see that when the light is on, we have polymer growth occurring. We turn the light off and it almost goes back to being nothing. We turn the light on and we get this nice stair shape going on, which is indicative of polymer growth in the light. And when we turn the light off, we can actually stop that polymer growth in its tracks. So we have very good control over knowing when and how we're growing the polymer. And these two data here on the bottom, these two graphs show us that we have molecular weights that are very low. So if we look at the right side in blue here, this is the polydispersity index. And all of the blue spots here, these are the polymers at different growth conversions are much lower than 1.1, which tells us that that distribution is very, very narrow. So if this was done by free radical polymerization without the RAFT agent in the mixture, these values would be up here, maybe even above 1.5, closer to 2, which tells us that the distribution, instead of having many molecular weights of polymer chain very, very close to each other, would be very broad like this. And so with free radical polymerization, you have some polymers that are very, very short chains and some that are very, very long chains. We can also show that the change in molecular weight, so these red dots, is in a linear process, again, as you'd expect through this controlled chain growth. And here is just the gel permeation chromatography to show us that the polymer, as time increases in size as we'd expect as well. So the nice thing, the reason we wanted to use quantum dots wasn't because we had great control over the polymerization. We wanted to use these for a recycling process. And so one of the major problems with using photocatalysts in polymerization methods is once you have this very small molecule as a photocatalyst, it's difficult to remove that from this polymer sort of product that you've produced. So we wanted to look at a way that maybe could we have the quantum dots, we could add monomer to these quantum dots in the solution, we could polymerize them and then remove the quantum dots and the polymer away from each other. And that way we could recycle and reuse the photocatalyst. And indeed, this works really well. So if we look at this schematic here, we have our vial, which has the quantum dots, the monomer in it, we shine light on it, we get the polymer. We can then centrifuge these out, keeping the quantum dots and collecting the polymer because they're of vastly different sizes. The quantum dots are about 10 to 12 nanometers in size, and the polymer is still on a molecular scale. So we can then centrifuge them out and separate them. We can then add new monomer and repeat this process over and over again. This is some UV visible spectroscopy here, which just shows us that this is the peak that we'd expect for the quantum dots in the system. And if we wash these 1, 2, 3 times, you can see this peak goes away very quickly, and we're actually able to remove all of the quantum dots from the system with only 3 washes, which is quite rapid. And then this data here is just a table that shows us that we can repeat this process, this recycling process 5 times using the same quantum dots to get the same pretty much molecular weights of polymer and very low, again, very low polydispersity. So this is really nice because if we think about what this might be useful for, if we think about how we could use this, this was the first example of using quantum dots for aqueous RAFT polymerization. So we're pretty excited that we're the first to do that. It solves a major problem that if we're trying to use this for an industrial process, the organic molecular photocatalysts that people used to use previously, they're really hard to purify out because they're on the same scale as the polymer and they can get entrapped within the polymer spheres. So the ability to be able to use that to then recycle those and reuse them is very useful. And this is important for a green production of polymers because we're using water here as the solvent. And so that's really useful in terms of reducing our amounts of organic solvents or chlorinated solvents that might be used previously to make these sort of materials. And so then my final story or talk that I wanted to bring to you today was looking at this, using this technique now, this PET RAFT. So this is using photocatalysts to do RAFT polymerization. But now we're trying to make a nanoparticle which is bioactive and using this method known as polymerization induced self-assembly. And I'll explain what that means on the next slide. But this was a large study that was done and led by a postdoc, a really talented postdoc, Hao Sun, who's in this picture here. He's actually got my daughter here on her first birthday, a really good friend of mine who we've worked together on this project. So I'm excited to see this finally get published. So the concept we're looking at here was we wanted to take a very special monomer, which I'll show you on the next slide, and make these hydrophilic peptide brush macro CTA. So what this really means is here we have a monomer that has a hydrophilic peptide attached to it. And we can then polymerize these hydrophilic peptides into a polymer chain. So we now have this polymer here that's very hydrophilic, and we can then polymerize this again off the end of it with a monomer that's very hydrophobic. And in doing that, when we have these in a water-based system, these hydrophobic monomers, as you can see from the schematic, want to pack into the core of the particle. They want to collapse in it and avoid the water that's around them. Where this component, which is made mainly from peptides, they want to then stay out in the water phase. They want to stay out and present. So we have these sort of diblock molecules where one portion is very hydrophilic and one portion is very hydrophobic. And that means they want to pack in and actually collapse in very well. Hold on, I just got a question here in the chat, so I can answer that. Sardeep Roy, he asked if can graphene quantum dots yield better RAFT polymerization results than cadmium selenium quantum dots? I think it's a really interesting question, and we don't know, but I think I'd be very interested in trying to figure that out and testing that. If you work with graphene quantum dots, we can definitely talk more about how we could do these sort of experiments and see how well they would work. From one thing, that would be good to be able to reduce the amount of cadmium and selenium, obviously cadmium being very toxic as a heavy metal. So if we get away from that and use graphene quantum dots or carbon dots, as some people are working with, that could be very interesting as well. Thanks for the question.
So we will take the question at the end of the lecture, Tristan.
Yeah, no problem. Okay, sure.
Okay.
So this is now made. We make this molecule here using this photo process. So this, as I described in the last study, where we use the quantum dots, now we're just using an organic photocatalyst now, but we're able to shine light on these molecules to make these without temperature, in just room temperature conditions. So really discussing this polymerization-induced self-assembly, this schematic here shows this process very well. So if we have a polymer chain here in blue, it's soluble, and if we think about this being very hydrophilic in an aqueous system, so this blue polymer here, we can then do polymerization from the end of this polymer chain using a monomer in red, which is hydrophobic. And so the hydrophobic polymer wants to then pack with itself and avoid being in this water space. And we can see, depending on the ratio, so the length of the blue polymer versus the length of the red polymer, depending on the size of those and the ratio of those, we can get these to pack in different ways. And by packing in different methods, we can change the structures that we can get. So here they're talking about increasing the core degree of polymerization. So essentially, if we made the red chain, the hydrophobic chain, longer relative to the soluble portion, we can go from these sphere-like structures to elongated worm-like structures, and then into more vesicle-like structures as well. And what that looks like by TEM, if you look here at these are all made by the same polymer, two different polymer components, but all they've changed is they've actually increased the length of the hydrophobic core polymer. And they can go very clearly in TEM, you can see from these spheres to these long structures, to then vesicles where you can see they're hollow on the inside, to these yolk shells, so there's sort of like a bit of a mixture of this one, to then these multi-layered vesicle structures as well. And this is quite neat because all we're doing here is we kept the same amount of polymer chain of the hydrophilic block, and we're just, again, increasing the polymer of the hydrophobic block. So in this study, we wanted to do this and use this to our advantage. And again, this is the same schematic, but you can see here, this now has a hydrophilic peptide. So this green represents a peptide sequence known as KLA. So this is a repeated KLA, KLA, KLA peptide. And what that has been shown to do is be very useful in being pro-apoptotic. So it's actually able to kill cancer cells. When it gets internalized, it's actually able to disrupt the mitochondria of the cells, and in doing that, cause programmed cell death. So what we did was we were able to polymerize these hydrophilic polymer chains with these peptides off the surface of them. And then when we added a hydrophobic monomer, you can see in the schematic that they want to pack in and make the core of the structure and present this peptide very easily on the surface of that structure. And so what does that look like? This is, first of all, just looking at by changing now, we have this same length of the hydrophilic peptide portion, and then we're changing the length of the red, the hydrophobic portion that's going to pack. So if we look here at these three different traces, you can see that the first portion is the same length, but here we now have 100 monomer units, 200 monomer units, and then 500 monomer units of the hydrophobic core packing molecule. And you can see very nicely by dynamic light scattering that the polymer actually changes the size of the particle very well. So here they sort of sit around sort of, this is about 30 nanometers to about 50 nanometers and now to 100 nanometers. So the size of the particle grows very easily. And this is the GPC trace, which shows us that the polymer is also changing, the size of the polymer is changing very uniformly. We can see this very easily by TEM. So here we have 5 different nanoparticles, nanoparticle 1, nanoparticle 2, 3, 4, and 5. And all we've done here is increase the length of that hydrophobic core. And you can see that the particles get bigger and bigger and bigger and bigger as we do that. What's nice about this as a system is this is all done by self-assembly. So these particles actually collapse and make themselves just through trying to control the surface energy around the particle. What was most important for us, though, is if we look here at circular dichroism, so this is now analyzing the KLA peptide sequence and then looking for that peptide sequence on the surface of the nanoparticle. we can see that the actual, the alpha helical structure, this is this double minima here, the alpha helical structure of the KLL peptide doesn't change once we add it to the surface of the nanoparticle. And that's really important because that's what gives it its proaptotic ability. It remains in an active secondary structure. So by polymerizing this and putting it on a nanoparticle, we're not altering what its biological function would be. What was more interesting to us was that we now have these nanoparticles where we have these brushes of peptide. So this is the KLA peptide on the surface. What we wanted to know is by tethering this peptide to the surface of a nanoparticle, does that improve its stability against different enzymes which might degrade these peptides in solution? And so here we're looking at challenging these where we add trypsin, which is an enzyme which can degrade peptide linkages. And we can then see that when we have the peptide on the surface, it wants to break up into these two major fractions. So we can see we have this 458 gram per mole fraction and this 329 fraction as well. So when we looked at this by HPLC, you can see here, this is now just the peptide alone. But after 1 hour of trypsin treatment, we can see that we've degraded all of this peak and now it's shifted to these two smaller peaks out here. And these two smaller peaks represent these two fractions that we're seeing in this case. And I'll show you some LCMS data of that. So we can see here that the fractions after just the peptide alone, after 1 hour have all gone. In the case of our nanoparticle systems, it actually takes out to 8 hours. We can still see presence of this original peak in this case. And so if we were to quantify that and show that all on the same graph, we're now looking at the percentage of cleavage of this peptide. So the percentage that this peptide is broken up into smaller fragments. The peptide alone, or the peptide when it's just on a polymer chain, but not actually in the particle system, we can see that it's very rapid, almost 100% cleavage of the peptide within 1 hour. Where the different nanoparticles, so we had different sizes of nanoparticles, 5 different sizes, we can see after 8 hours of time, they still remain very, very stable. So only 40% of the peptide after 8 hours is actually degraded. And so what this is telling us is that by tethering this and putting it on a nanoparticle, we're actually able to enhance the stability of these peptides against proteolytic degradation, which is very useful if we're trying to use these for a therapeutic benefit in being able to kill cancer cells. So now we're just looking at some cell biology data or in vitro data. If we look at the images first, for example, so now we're looking at no treatment in the top. This is a control set here. When we add just the peptide alone, this is a control molecule. This CCP is known as, it's a carbonyl cyanide 3-chlorophenyl hydrazone. What this is pretty much a positive control. This molecule here has been known to actually disrupt mitochondrial membranes. And then we're looking at our nanoparticle for half an hour, and then after 2 hours of treatment. We have Hirsch stain for the nuclei of the cells. This JC1 reference here. So JC1 is a molecule that in a monomer state will stain mitochondria. So here we're looking at all the different mitochondria in these bars. But in healthy mitochondria, it then wants to form these aggregates which show up in the red channel. And so you can see when there's no treatment, we have lots of healthy mitochondria. In the peptide alone, we have lots of healthy mitochondria. Even in the CCP, which has been shown to disrupt mitochondria, we can see some evidence of that. But in our nanoparticle case, we see we're really disrupting all of the mitochondria that's present in these cell structures. And you can see by the bright field, the cells are clearly disrupted and starting to die and become apoptotic. When we then quantify this in terms of actual cell viability, you can see here that as concentration of the nanoparticles increase relative to the peptide, we see a large decrease in the amount of actual cell viability we're getting with these. So these nanoparticles not only are able to protect the peptide from being degraded, they're also able to actually have enhanced effect in terms of their anti-cancer cell toxicity capabilities. And so this was recently published in Angueta Chemi, as I said, with Hao Sun as the lead author on it, a fantastic friend and collaborator of mine. And just wanted to highlight some of the advantages of this. So what's really nice is we're able to use this PISA, this polymerization-induced self-assembly process, to make a nanoparticle which has some therapeutic function and benefit. I think is really exciting. the improved peptide stability is something that we're really excited about. And this is something that isn't just unique to this peptide or unique to this nanoparticle system. There are so many different peptides we could look at and try in this same process to see if we could improve the peptide stability. So we're really interested about that. And the idea of being able to target different hydrophilic therapeutic peptides for not just cell death, but to look at ways we might be able to use these for tissue regeneration or for different applications is something I'm pretty excited about as well. And so that was the last story I had to present today. So as I said, 3 different projects from 3 different groups, essentially. The first project coming from my work back at University of Western Australia with I.S. Ramanathan as my supervisor. The second project was in collaboration with the Weiss group and this PhD student who's just finished, Kevin McClellan. And as I said, project 3 was a large study. We've just finished here in the US as well with the Gianeschi group and my supervisor, obviously Sam Stoop, and Hayo Sun, the lead author on that. These are the funding bodies that supported me with my work at University of Western Australia. These are the funding bodies supporting me here at Northwestern. And I just wanted to highlight this video. This was one of the reasons I've been back to India a few times is with the Rotary Foundation to deliver microscopes. So this is a program I'm a part of called Microscopes in Schools. And we were able to take some microscopes out to some village schools just outside of Chandigarh and deliver these microscopes, so the students had an opportunity to be able to see and see what's happening on the micro scale a little bit closer to the nanoscale that I work on so much in my research. And a very special thanks to Professor Sinha and obviously to my PhD student that I'm co-guiding back there in Amity in Pune. I'm hoping I can talk to you today on this call as well if you're online. And obviously her other co-guide in Suman, Professor Suman. I've been very happy to be able to work with all of you in this project. And so now I'll finish and happy to take any questions with Professor Sinha. Thank you so much.
Thank you, Dr. Tristan, for really a wonderful talk, and you have shown that how these polymeric materials and the polymer nanoparticle can be used in the biomedical application, especially the cancer therapy and others. So now the session is open. Anybody who want to put the question, either they raise the hand, we will be unmute you, and then you can put your questions, or you just put your question in the question and answer session. So we will do that. So those who wanted to speak, they can raise their hand. We can unmute you. Meantime, you have some questions and answer session. So already Dr. Tristan has given the reply to the Swaradip. We have another question there, sir. How does the RAFT agent be removed, and how long can RAFT keep the polymer dormant?
Yeah, so it's a really good question. The RAFT agent can actually, there's two parts of that question. So the RAFT agent keeps the polymer dormant during the polymerization process. So it's actually very short amounts of time that it keeps it dormant for. But if you think about that, just regulating the addition of monomer through that time. So it's very like nanoseconds that it's dormant for. But maybe you're also asking about how long can that RAFT agent stay available to grow polymer with? And that's a really good question, because as long as that RAFT agent is still attached to the polymer chain, you can purify these polymers, you can freeze dry them, collect them, and you can then add more monomer and photocatalyst and then reactivate from that polymer chain end. So as long as that RAFT agent is still in that dithiocarbonate structure, it's still active and still available to be used. And then maybe the final part about how can the RAFT agent be removed. In this project, we didn't remove the RAFT agents, but it's very easy to do that if you wanted to. You can use a range of different techniques now. The easiest one being addition of a different radical, which can then cleave that, for example, from the end of the polymer chain. The other nice thing about using RAFT polymerization relative to free radical polymerization maybe is that you have this active end group, which is a dithiocarbonate. You can then do chemistry on that to then have chemistry or different additions to the very end of your polymer chain, which is difficult to do with other polymerization techniques. So yeah, thank you. That's a good question.
So another question is by Swaradip again. During the photo excitation of the electron in quantum dots and eventually transfer to RAFT agent, does the quantum dot band gap play a vital role in efficient electron excitation and the RAFT process?
Yeah, a really good question. So essentially, yes, it does. The band gap has to be high enough to actually be able to fragment the RAFT agent. And so I guess the activation energy or the energy required to fragment the RAFT agents is different depending on which RAFT agent you're using. But as long as the energy transfer from that band gap is large enough to make that happen, then this process should work. So in relative to graphene quantum dots, provided that band gap energy is large enough to be able to then fragment the RAFT agent, then that should be okay. Yeah, and obviously then depends on what input light you want to use as well.
So another question again related to that, can RAFT be used to break down polymer using the light-induced process?
Yeah, it's a good question. Can RAFT be used to break down polymers using the light? Yeah, so there is recent work, and this is something that's pretty exciting. I don't have a lot of expertise in this, but being able to use RAFT polymerization with different photocatalysts or different catalysts in general to actually depolymerize polymers. When you think about that from a recycling point of view, it's something that's very interesting and very new in the field. They're only starting to publish a few papers on depolymerization using RAFT. In saying that, using light without a photocatalyst, for example, these are still carbon-carbon polymer chain backbones. So it's very difficult to break them up just using light as the process for that. So they're not really biodegradable in light, if that's kind of where the question was asking as well.
Neeraj, can you unmute Poonam Mehandia? She put the question, but since Dr. Tristan is the guide, co-guide of her, so we can unmute her and she can put her question. Although she has put the question in the chat, but I want that she should speak. Can you unmute?
Me too. I want to hear from Poonam as well.
Najir ji? Hello. Hello. IT support? Okay, probably the IT support is not around. So now the question by Poonam Mehandia is that what is the biocompatibility and toxicity level of fluorescent dye and the magnetic nanoparticle?
Can you say that again, Dr. Sinha? Just I missed that.
What is the biocompatibility and toxicity level of the fluorescent dye and the magnetic nanoparticles?
Oh, okay. Yeah. So this is referring to, I guess, project one, I guess the nanoparticles we made in that case. Yeah. So the, a few things. So the nanoparticles we've shown in other studies, I didn't show the data today, but the magnetic fluorescent nanoparticles are very biocompatible. We can add these to primary cell cultures. So central nervous system cells like neurons, for example, up to a concentration of 10 mg per mil. So 10 milligrams per mil, which is very high for these nanoparticles. What's more important, though, I think, is that we can show that these nanoparticles can be cleared very easily from the body. So we've done a cancer-based study with these nanoparticles and showed they didn't accumulate in any organs so that then cause toxicity or any problems. And we've also shown that they can be cleared from a spinal cord injury model using the optic nerve and showed that after 2 weeks, we couldn't find the nanoparticles in any of the organs. So they're small enough, they can be cleared by the body and they're not accumulating in any vital organs, for example, to cause some toxic effect. So yeah, great question.
Again, he wrote, thank you, sir, for wonderful session. There is another request, basically. What about the project 3 slide PPT? Can we get that? So as I told in every this presentation, these are the IP rights of the presenters or the person who is speaking. So it's better, I don't know if Tristan is happy to share, that's fine. Otherwise, these are the copyright of the presenters. So we cannot ask anybody that you want to share or not. So Tristan, somebody want the project 3 slides?
Sure. Yeah, I would just say, I mean, today I haven't presented on anything that's not published. So so this all the details for project 3 are in this paper. So if you wanted to write down this reference, this was literally only published like last week. It went online, so it's just online now on the website. I'm also aware that maybe sometimes it's difficult to get papers. So if you want, Professor Sinha, what I can do is I can send you the PDFs of the papers, and then you could then forward them out, for example, if you wanted to, because I understand sometimes some libraries have different access to different journals and things like that. So that's no problem. I'm happy to share the 3 papers of the 3 stories I discussed today, and I can send them to you, Professor Sinha, and then you can send them out to anyone that requests them. That's no problem.
Yeah. So basically, I request the attendee that put your name. I am getting the question by anonymous attendee, so I could not know who is asking. So another question is that, can the photocatalytic keep up the bulk reaction? Keep up in the bulk reactions?
Yeah, so I guess that's referring to using the photocatalysis in, I guess, a bulk polymerization. So no solvent, I think they're asking. So just total monomer. And yes, they can. They've used that before. It does become a problem to try and make high molecular weights in doing that, mainly because you then start to have viscosity problems and sort of the ability for the photocatalyst to be able to move around in those systems. But they can make ultra high molecular weight polymers using this, mainly in organics, so not in aqueous systems. Yeah.
There is another question by Poonam Mendia from Kurukshetra University, and she is the one of the author of the paper which we have accepted recently.
Sure,
so as the size of the quantum dots tune the band gap, and so what preferably size of the chosen, and also if we go from in vitro to in vivo, what are the scaling parameters to take care? So first question is that since size tune the band gap, and what side we should choose, number one, and with the moment we are going from in vitro to the in vivo, what scaling parameter we should take care?
Sure. Yeah. Okay. So I think so. Let's talk about the first part with the quantum dots. We're using green light excitation for our quantum dots. So the size of the particles, if I remember correctly, it was about 10 to 20 nanometers in size to achieve that with cadmium selenium. One thing that's very interesting is that for RAFT, and if we're thinking about new projects or new ideas, for example, is that they're able to show now that different RAFT agents can be excited by different wavelengths of light because they have different activation energies to be able to fragment them. So the ability to have maybe 2 different quantum dots in solution of different sizes, of different band gaps, which could then activate 2 different RAFT agents, could be really interesting, because then you could actually selectively, depending on the wavelength of light, activate 1 quantum dot, which can then activate 1 RAFT agent, and then change the wavelength of light and activate a different quantum dot to activate a different RAFT agent. And being able to do that all in the same system could be very useful to make different polymers or different types of polymers, for example. To the second point about the scaling, I guess, from an in vitro to an in vivo system, it's a really good question, because if we talk about project 1, the polymeric nanoparticles we made in that system, that's a very batch to batch dependent process. And that's a problem for us. We can really only make those particles on a scale of about 30 to 40 milligrams per batch, and that's quite low. Project 3, however, the polymerization induced self-assembly system, that's a process that could actually be scaled to a flow system. And in saying that, we could then make that on a much higher scale to hundreds of milligrams, maybe even to the gram scale. And the ability to be able to control that and then use that in a scaling from in vitro to in vivo wouldn't be as big a problem. So I would say a great question. Project 1, difficult to scale. We could scale it to small animal models, but to really scale it to sort of large animals would be a challenge. Project 3 would have no problem with that because of the way we make the particles.
I have some, I just need your suggestion. Suppose I have such kind of, because you are showing the large scale micron size particle, the polymeric nanoparticles and those things. Suppose I trying to use the different kind of the radiation source. For example, I can go for the synchrotron radiation and you go for the CT imaging, right? So is this nanoparticle, or this particle which we are getting from the fluorescence spectroscopy, are we able to get some data from that also? This is my doubt only. It may not, but what is your comment about that? Can we use the nanoparticle for the CT imaging using the synchrotron radiation or those kind of things?
Yeah, so I mean, we focused in the first project primarily on using them for magnetic, so MRI imaging, as well as fluorescent imaging. but there's definitely a lot of work, and the potential to encapsulate a hydrophobic probe for CT imaging or for radio imaging, for example, is very possible. We haven't done that yet in the lab, but it's something that other groups have done, and with very similar particles, we could probably make something like that work. So as long as we could encapsulate that or chemically or covalently attach that to the nanoparticle, then I see no problem with being able to make that happen. Yeah.
So the moment we are putting the nanoparticle inside the cells, there should be a change in their electron density, or maybe some density of the material.
Mm-hmm.
So
yeah.
is some kind of the observation you have observed, or you are just from the microscopy, since there is a contrast changes, there we are saying that it is.
So with when the nanoparticles we've seen, when they get internalized within the cells, they normally get packaged up into endosomes and lysosomes. So that takes the particles into, I guess, aggregates or agglomerates of those particles within the cells. So there is definitely a concentrating effect that they then get packaged into these vesicles, of many nanoparticles before they're cleared from the cell. So I would definitely say our evidence supports that hypothesis that they are getting bundled together and I would say attracted to each other by the cell. They're getting packaged together.
Yeah. So there is another question by one of our undergraduate students. Can the hydrophobic and the philic part decreases activation energy in the PISA way of polymerization?
Hydrophobic and philic part. It's a good question. So there's a few, I guess, probably it sounds like you're asking about the activation energy for the polymerization to grow, I think. And it is very, the nice thing about RAFT as a polymerization technique is that they're very widely available to a range of different monomers. So they can actually polymerize charged monomers, anionic, cationic, hydrophilic, hydrophobic, different monomer sources. Doesn't change too much. There are some monomers that are difficult to polymerize by RAFT, and that can cause a problem because of the mismatch between the fragmentation of the RAFT agent and the activation energy to actually start a propagating chain of polymer with those RAFT agents. But in terms of our study, what we saw, we didn't see much difference in terms of the monomers we used and the activation energy or the propagating energy for the hydrophilic and hydrophobic parts. So I would say that globally, answering your question, that there is differences between monomers, so hydrophilic versus hydrophobic monomers, but RAFT is very good for a high range of monomers that we don't really see problems too often.
Yeah. Probably the PISA, Chinmay, it does mean by polymerization into self-assembly, you are talking about the PISA? Chinmay, can you type another that if it is polymerization into self-assembly, yes, the activation energy play a role.
Oh, yeah.
Yeah, so, okay. So the same thing he is asking about. So already, okay. Yeah. So Chinmay has, that is there. So any further question from the student side? A student, faculty? So thank you. Another question, I think. No, it is okay. Only Chinmay is there. So thank you, Dr. Tristan, for a really wonderful talk, and especially let us know that there is a different way for the polymer nanoparticle to use in the biomedical applications. So thank you very much, and we hope will be still collaborating in the future also. And I would love if some of the students can contact you for the either dissertation work or the PhD work in either Australia or the USA. So thank you very much on behalf of the Amity Institute of Nanotechnology and the Amity University, Uttar Pradesh, and the management. I am really thankful to you to give the time. Although you are, whenever I am asking, you are always available, I know. But again, whenever you are visiting India, please spend some time with us. Maybe we will be arrange the accommodation and other things at our Amity University, and maybe we will go for some series of the lecture for that. So,
of course. Yeah.
So we will see that. So thank you very much. Thank you all the audience, and thank you students for joining today. Thank you all. Thank you, Munir, for the IT support. Thank you.
Thanks so much.
Thank you.
Thanks. Bye.